Good afternoon. First of all, I would like to thank the organizing committee for giving me this opportunity to present you our results. And I will start with a few words about my country, since probably not all of you are familiar where is it? It is in a Balkan peninsula. It is landlocked country, southeast of Europe. About 60% of our country is arable. And today, agriculture is basically our only element of economy with a positive international trading balance. So it is extremely important for us. In my talk, I will first shortly introduce you to the subject. Then I will describe the organization of our research and sample collection, and I will focus on morphological and molecular identification because we were trying to find adequate taxonomic criteria for proper identification, which is a frequent problem within the genus of Alternaria. Parsley is, as you all know, specific plant, herbaceous plant with multiple usages. Leaves are used as a spice, taproots as vegetable, and it is rich in essential oils used in medicine or cosmetics, so it is considered to be important. But on the other hand, worldwide, it is considered to be a minor crop with production on limited scale. But in Serbia and probably elsewhere, it is present in our diet on a daily basis. So probably it is more important that it could be estimated only based on value or areas for its production. Among a lot of pathogens which could endanger production of parsley, genus Alternaria, especially Alternaria petroselini, is considered to be the most important, causing black leaf spot, and there are several other Alternaria species of undefined taxonomic status also described to cause this disease. Alternaria petroselini has very complicated pathology, epidemiology. Could be seed-borne, soil-borne. It is long persistence in the soil, and it probably has narrow host range, mainly including, besides parsley, parsnip, celery, and fennel. There is no much data in literature on Alternaria petroselini, but it is described to be present in several parts of the world, including several countries in Europe. In Serbia, it was first detected in 2005, and it is present in higher or lower incidence ever since. It is placed taxonomically within Radicina species group, and it is very closely related to Alternaria radicina and Alternaria carotinsulte. And because of overlapping morphological and similar morphological feature, the identification could be quite difficult. So the main goals of our research was to examine main morphological features in order to find adequate taxonomic criteria, to establish experimental host range, and to test suitability of several, namely 5 genome fragments to be used in proper molecular identification. We were performing sampling from 2005 until now, and we were using different sources as plant tissue, seed, soil, by using baiting plants, and use conventional mycological methods. In the field, symptoms usually appears with as small necrotic spots, and pretty soon, from the edge of the leaves, necrosis starts to spread, and in several days, the whole leaves are collapsing. As you can probably see on these leaves, Alternaria petroselini is present usually in a high incidence. Almost all the leaves are affected, so these plants are completely unmarketable. Our isolation was very successful from several sources, leaf spots on the left, from root neck of tap roots with a canker. From in the middle, you have baiting plants from capturing Alternaria petroselini from soil samples. And at the bottom, you have a petri dish with seed, parsley seed, and around which are conidias of Alternaria petroselini. We have obtained over the years a total of 19 isolates, and we performed their detailed characterization. Morphological features that we studied, including macroscopic features as colony appearance, formation of pigments, crystals, and microsclerotia, while microscopic features, including all elements of sporulation and conidia appearance. Isolates form dark gray or dark brown conidias with specific glow, as you can see. And as the opposite of Alternaria radicina, as I said, which is closely related, Alternaria petroselini has fast-growing colonies and does not form any crystals or radicinin in a culture. Conidia were formed. Sporulation was abundant, and conidia formed on a short, unbranched conidiophorus. And this is direct sporulation of Alternaria petroselini on parsley seeds, since we have 14 isolates originating from seed. Conidia are ellipsoidal, darkly dark, but either sporulation pattern and conidia appearance could not differentiate Alternaria petroselini from Alternaria radicina, so this is not proper taxonomic, this proved to be not a proper taxonomic criteria. Nevertheless, all Alternaria petroselini isolates formed this microsclerotia beneath agar surface, and this proved to be very specific and valuable taxonomic criteria. Although this species was Alternaria petroselini described a long time ago, the presence of microsclerotia is first time discovered in 2002, and after that a lot of other authors reported that their isolates for microsclerotia. Pathogenicity tests include artificial inoculation of 7 Apiaceae and 5 known Apiaceae plants by spore suspension spraying. And first of all isolates cause the appearance of necrotic spots on parsley leaves 6 days post-inoculation, and thus all isolates confirmed Koch postulates. Experimental host range proved that all inoculated plants could be divided in 3 groups. Alternaria petroselini was highly pathogenic for, besides parsley, for parsnip, celery, and coriander, weakly pathogenic for the rest of Apiaceae plants, and non-pathogenic for non-Apiaceae plants. Here is the reaction of inoculated plants: necrotic spots on a parsnip, similarly necrotic spots on a celery leaves, and finally this is the reaction of coriander to the inoculation with Alternaria petroselini. Finally, we have tried to use to find out the suitability of 2 different specific primer pairs, designed for Alternaria radicina, but described to amplified Alternaria petroselini as well, to see if they can be used for delineation. And molecular identification was performed on the basis of sequencing this named 5 genome fragments, 2 fragments of ribosomal DNA, ITS, and small mitochondrial subunit, and 3 protein-coding genes, Alternaria allergen 1, beta-tubulin, and EF elongation factor 1. Specific primers amplified all the isolates of Alternaria petroselini, but all the isolates of Alternaria radicina as well, and the difference could not be made, so the use of these primers for species delineation is not possible. Further on, molecular identification was performed, and thus we have developed, we have sequenced 5 genome regions, and within, among 2 Serbian isolates that were sequenced, we have found, as we expected, no variability in 3 genome fragments, but we think it is very valuable. We detected variability, namely 2 nucleotide substitutions in a sequences of 2 protein coding genes, and this is the first evidence of intra-species variability of this species. Among all 10 generated sequences were used for blast comparison with all sequences in GeneBank database, and it showed that our sequences exhibited 93 to 100% of nucleotide identity with other Alternaria species from other parts of the world, but with several closely related species, and there is a huge difference between suitability of tested genome parts for to be used as the taxonomic criteria. As most useful proved to be Alternaria allergen 1 gene, showing that Alternaria petroselini has 25 to 27 nucleotide substitutions of difference between comparing to all other closely related species. And useful was also EF1 factor with 12 nucleotide substitutions. Informative but not completely decisive were beta-tubulin region and ITS regional ribosomal DNA with 3 to 5 or 5 to 6 nucleotide substitutions. On the other hand, a small mitochondrial subunit proved to be conservative within this, within a wide range of Alternaria species and Ulacladium, Stemphylium species from other genera, so this genome fragment cannot be used for molecular identification. And at the end, we have concluded that among tested taxonomic criteria, fast-growing colonies and the presence of microsclerotia, coupled with the sequences of Alternaria major allergen 1 and EF protein coding genes are reliable and should be used for species delineation and identification. Furthermore, we have detective interspecies variability of our isolates compared to other closely related species in 4 genomic regions, and we have detected intraspecies variability in 2 genomic regions. We have also defined future goals for our future investigation, and it is to establish reliable and suitable molecular protocol for detection of Alternaria petroselini in parsley plants and seed, and to continue to explore genetic structure of population of Alternaria petroselini. And at the end, thank you for your attention, and I'm ready to take your questions.
Pascal Poupard. Did you use other molecular markers for intraspecific variability, such as intergenic spacer, or which are more variable than ETS?
The species delineation between Alternaria petroselini, radicina, and carotinsulte is problem which is going through the literature for several years, and Professor Pryor from US has done a pioneer work by recommending several regions that could be tested. As far as I know, intergenic regions was not tested for Alternaria species delineation, and there is no available sequences in a gene bank to compare your data to, so we didn't use it. We just used this. We used some extra protein coding genes which are not presented here, but no, we didn't use intergenic region.
Because it is relatively easy to use.
Yes, I know, but there are no sequences to compare to, because if you have first sequence, you cannot do anything with that. Yeah.
Thank you.